Similar question with a little detail switch. So if you study the problem sets and you know the answers for the problem sets without your notes, you'll be good.
All right, and with that, we'll probably get started. Okay. So, welcome in. I'm Angeli.
I'm Yago.
Gonna go through again all topics today. Probably gonna focus a tiny bit more on 2 and 3, just because that's the most recent stuff. We have midterm questions, the mini midterm questions. We have some problem set questions. We're gonna go through all of it. Again, question, raise your hand. If you interrupt me, I will totally forget what I'm talking about. So, yeah, let's just get into it. Okay. Okay, topic 1. So again, this stuff, pretty straight out. Not going to go too crazy with these questions, but we're going to go over some key things. So differences between prokaryotes and eukaryotes. Prokaryotes, no membrane-bound organelles. That's probably the biggest difference. But let's talk a little bit about the main difference between these two, which she's asked you guys about before. So let's just start with the first one. So transcription/translation occurs blank in prokaryotes. Yeah. It occurred. Okay, so it's either separately or simultaneously. No, it's okay. Excitement. We're tired. Do you have a guess for that? Do you want to try it, that one? You said simultaneously? Okay, and who can do the second one? Well, transcription/translation occurs blank in eukaryotes. It's the other one. You can say it out. You can say it out separately. Okay, and who can tell me why? Why are one of them separately and one of them simultaneously? Yeah. Space and time. Transcription, translation, these all take time. And they also occur at different parts in each type. So who knows? So simultaneously, yes, they occur. And he said in the back, where do these two processes occur in prokaryotes? Where? Cytoplasm. Both of them, right? Same time. Because there's no need to like go somewhere else to go and do the second thing. So what about eukaryotes? Where does transcription happen? Nucleus. Okay. And what about translation? Cytoplasm, right? So you're exiting and you're going out. That takes time. Does that make sense? Pretty basic. Okay.
Yeah. All right. So the central dogma of DNA, right? Whenever she talks about this, she's talking about how we get from DNA to proteins. We know the first step in that process is transcription. That's when the DNA code is read and it's turned into an mRNA code, right? Where do we think this occurs and what's it done by? What part of the cell does it occur in first? We'll talk about eukaryotes first. Exactly. So this happens in the nucleus, right? Because that's where the DNA is found in cells, at least in eukaryotes. And it's done by the RNA polymerase. Think RNA polymerase makes RNA. All right. So what you prefer those?
Here, I can do this one. Okay, second one. Blank, guide blank to know where on the promoter to latch onto. This is like super key. You don't want to forget about these two terms. Who wants to go for this one? Blanks, guide blank to know where on the promoter to latch onto. He said one of the terms just now. I know you know. Anyone else? Anyone else?
What guides the tool that makes it?
What's that term? What's that term?
Transcription factors guidelines.
Yes, transcription factors. Transcription factors latch onto the promoter, and it's telling RNA polymerase where in the gene to go to do its job, which is to make DNA into mRNA, right? So without that, like, think about how long a piece of DNA is. Like, we talk about little short strands here, but they're like hundreds and hundreds of like nucleotides long, right? How do they know where to go? They know because of the transcription factors. And let's do the last one super quick. Both DNA and RNA are made out of what? I just said this term. Nucleotides, right? And what's the difference between those two? Yeah.
RNA has an extra oxygen while DNA does not.
Yes, that's the structure, but I'm thinking more nucleotides. Yes. Right. So DNA has the T, that T base. RNA has that U. So part of your exam, let's say she asks you if it's like double-stranded, single-stranded DNA or RNA. I'm going to know it's RNA straight away if I see a U there. Okay. So just to recap, occurs in the nucleus, done by RNA polymerase, right? Just like Yago said, RNA equals, they're making RNA. Transcription factors guide that to know where the promoter to latch to. And then we know that both of them are made of nucleotides, and that difference is RNA has the U, DNA has the T. Okay, let's do translation. So think about it this way. Transcription is DNA to mRNA, and I think of translation as everything else, right? So mRNA turned into the amino acids, right? And then it goes, you put a bunch of them together, and you get a protein. Okay, and if you remember, the types of amino acids, like different types of amino acids that are strung together, because they're chemically different, are going to adjust the protein folding of that protein that's made. So if I switch out a bunch of amino acids and I replace them with different ones, it's going to fold differently because they're different chemically. And what is that phrase? What is that phrase she's always saying? That she's always saying, structure determines what? Function. Okay. So where is translation occurring in and who does it? Occurs in the cytosol, done by what? Ribosome. Ribosome makes proteins. Got it? Ribosome makes proteins. What RNA? What type of RNA makes up the ribosome? rRNA. So pretty straight up. rRNA makes up that ribosome. Okay, cool. Any questions on this? We'll get to that. We're gonna do a whole thing on that. It's 3 to 5. So like transcription, RNA polymerase starts at the 3' end. It's moving its way to the 5' end. And we're gonna get into all of that in a second, just how that works. So that's transcription. Translation, that's a whole different process. So we'll talk about that in a bit. You want to do this?
All right, and then there's the different types of macromolecules that you guys have to know. So we have amino acids. Can anyone tell me which one is the amino acid? Yeah, exactly. So this right here is the amino acid, right? Every amino acid has the same basic overall structure, or the backbone, has the carboxyl group right here, the carbon with the oxygens, right? That's why it's called carboxyl. It's got the amino group. Think about the N in amino for nitrogen. And then it's got the central carbon, always bound to a hydrogen. And then the difference between every amino acid will be the R group. What about this? Does anyone know what this one is? Yeah, so those are nucleotides all together. Specifically, this one would be RNA.
And how do we know it's RNA? Because we see the U, right? And do you know what, when you put a bunch of stuff together in a string, what is that called? Polymer, right? And the things that make those up are called what? Monomer. So in this case, with the RNA polymer, what is the monomer? What are they made up of? Nucleotides, right? So putting a bunch of nucleotides together makes up the polymer, which is the RNA polymer in this case.
Okay, now moving on to this one. Does anyone know what this one is right here? It's a nucleotide, right? So again, nucleotides are what make up DNA or RNA. Do we know if this would be an RNA or a DNA nucleotide? Why? Yeah, right. So it's got the two OHs right here. RNA, just ribonucleic acid. DNA, deoxyribonucleic acid. So it's missing an oxy group right here.
She had a question.
No. So going on with that, what part of the nucleotide do we think this is? What do we call this? The base, right? This is the part that's going to be different in all the nucleotides. So this is the letter code and what matches with the other one on the opposite strand. What about this part? the pentose sugar, that's the same in all the nucleotides, different in DNA and RNA with this group right here, but otherwise the same. And then what about this part right here? That's the phosphate group. And notice it's negatively charged. That might come up. All right. So DNA or RNA is always negatively charged. What do we think this is? Phospholipid, right? Whenever you see something with long fatty acid tails like that, always phospholipid. Another hint, phosphate. You guys see the phosphate group right here, similar to the nucleotide. Same thing. Do we think that would be hydrophobic or hydrophilic? Or both? And what parts? Yeah, it has both parts. So what do we think this tail, the fatty acid tail, would be? Hydrophobic or hydrophilic? Hydrophobic, right? It's scared of water because it's got so many of these nonpolar bonds together. And then this part right here, the head, hydrophilic because it's got a bunch of polar bonds and that would interact with water.
And if you just think about the structure itself, like the heads are the ones that are actually touching like the aqueous solution, right? So it makes sense that they would be hydrophilic.
And then last one right here, what do we think this is? Carbohydrate. Perfect. What are some identifiable features that make it a carbohydrate? Yeah, but what else? Exactly. So when you see shapes like this, like the rings all together, most of the time that'll be a carbohydrate. Everyone good with this stuff?
Okay, fill in the blank. TNRA brings in the blank based on the codons present. Let's have some guesses. Get some hands raised. I know it's late. Let's just try to get through it. The faster we get through it, the more we can move on. Yeah. Amino acids. Great. So does anyone know what a codon is? What is a codon when I say codon? Yeah. Yes. So amino acids. Perfect. Codons are basically the groups of 3 of the nucleotides of DNA in DNA RNA that corresponds to a specific amino acid or stop signal. So that chart you guys have with all the amino acids, each of them is coded by 3 nucleotides. Okay, so depending on what that group is, like you can see this here, this is tRNA. Each group of 3 corresponds to a particular amino acid, and that also includes like the start and stop signal. Okay, so that's what a codon is. It's those groups of 3 nucleotides that tRNA is taking in to make stuff. But that's more like topic 4, but just so you know what a codon is. Okay. No. Okay, differential gene expression. This is super, super important concept. Basically, it just means that we're all made up of the same genome. We all have the same genome. The difference is in us, both physically, like I don't look the same as Yago, Yago does not look the same as me, is because we differ in what genes are expressed, okay? So, and this can differ based on a lot of things, but it also includes like the cell's environment. That's all it is. So basically, we all have the same genotype, which creates different phenotypes. Okay, so she ever asked you like, does one type of cell have certain information that another type has, like yes or no? They all have the same information. They're just choosing different parts to express and different parts not to express. Does that make sense?
So I talked about this analogy in the first review session. For those people that were here, the first review session, think about this. So you begin with DNA, right? And DNA is like, think of it as a cookbook in a language you can't understand, right? First step will be to translate it to a cookbook you can understand. That would be to mRNA, right? So you're translating the cookbook, now it's in English, and you can understand it. Now you're making recipes, which are the proteins, right? So transcription. Now you have all the recipes you can make, but you're choosing which ones to make. Translation, sorry. Now you have all the recipes you can make, and you have to choose which ones you're gonna make. So everyone has the same cookbook, but someone can make pizza and someone could make bread. you started with the same code, you translated it to something you can all understand in the same code, and you chose different parts to express. Does that make sense?
Anyone do this? You want me to do this one?
Okay.
Okay, so mini midterm question. Had a lot of questions about clarification when you guys are taking this. So we're gonna go through each one in this question. So this is what the question's asking. And by the way, this is exactly how you were graded. Notice 0.5 point for correct yes or no, 1.5 points for correct explanation. That is the great thing about Laura's class. Even if you directly answer it wrong, but it kind of seems like you understood it based off your explanation, you still get a lot of points. Okay, so yes, you're going to directly answer the question, but always explain rather than, let me just leave that out, okay? More explaining is always good than less explaining. So, consider a cell an organism like your beloved cat. If you were to isolate a cell from the cat's skin and one from its brain, you have a skin cell, brain cell, which of the following would be true of these cells? And for each question, answer yes or no, and then explain your reasoning. So, let's talk about the first one. The cells will contain slightly different DNA nucleotide sequences because the DNA nucleotide sequence is changed as cells differentiate. Okay, this is, you guys know this, okay? It's the wording. Don't let the wording trick you. You know this in your head. You know the basic principles. Let's just work through it. Anybody have any ideas? Or like, if you remember what you said on the exam, yeah.
The DNA nucleotide sequence itself in the cells don't change. It's the gene expression that's changing with how the cell acts.
Exactly. So they have the same DNA nucleotide sequences. Okay, because everybody has the same what? Genome, genotype, that thing. Okay, we all have the same thing. The only thing that's different between different types of cells and what makes us different from each other is which of those genes are being expressed. Okay, so that's a very key principle that she's changing the wording and trying to confuse you on, but you know this stuff. Okay, so if you're panicking on an exam and you're like, the wording is crazy in this, you know it, I promise, more than you think you know it. You really have to get to the bottom of the question and really just figure out what it's asking. Does that make sense? So yeah, so not true, right? No, generally when cells differentiate, the DNA remains unchanged. What's different is when the genes are transcribed and translated. So what changes? The genes are expressed. So we all start out with the same ingredients, recipe analogy, all got the same cookbook. We're all making different recipes, okay? Remember, different cells have the same genotype, different phenotypes, okay? Already went over that. So every cell, same DNA. I'm saying it over and over again because I really want you to get this in your head. The only thing that makes us different from each other is which genes we each choose to express. So we're all working with the same ingredients. Got it? All right, B. The cells will contain exactly the same proteins, but what would make the cells different in that some of those proteins would be active in the skin cells and some would be active in the brain cells. Even before we answer this question, straight off the bat, what word am I seeing in here that's going to make me suspicious of this question? What word? Exactly. Exactly. This is bio. So I always kind of say like, don't always like, never say no in a way. Like the cells will contain exactly. I said the word exactly, I'm sus. I'm super sus. Okay. So somebody help me answer this question. Cells will contain exactly the same proteins, but what would make the cells different is that some of those proteins would be active and some would be active in the brain cells. You can answer this for me. Just a guess. I don't care if it's wrong. That's why we're here. Anything. I know you know it. Anyone else? I know it's late. Help me out here so we can get through it. No. We're hearing no. Okay. No, it's not true, is what I'm assuming you mean, right? No, it's not true. Okay. Why? Why? Yeah. Gene expression. Once again. Yeah. We're here. So some proteins might be the same, but many would be different. Why? Because these are two different types of cells, right? Skin cells and brain cells. What makes cells different from each other? Gene expression. Cool. And then I think it's one more. Some cells would be expressing mRNA and others would not be expressing mRNA. Thoughts? Yes or no? Let's do a thumbs up for yes, thumbs down for no. Okay, no. Anyone can tell me why? Yeah.
Yeah, also need to utilize mRNA for gene expression.
It's that easy, okay? So wording weird, but you know it. And if you need like a little pep talk or something, and if you need some clarification, we can do that on the exam. Okay, you know more than you think you do. So, complicated question, easy answer. No, everything, all living cells need to express mRNA. That's literally the information that is within them in the cell. Okay, so genes need to be transcribed as proteins are constantly being made in cells. Okay, some cells are expressing mRNA, some would not be expressing. Maybe you're like, oh, well, gene expression, like maybe that's different, but they all still have mRNA. They're all still expressing mRNA. Just different parts of the mRNA are being made. Okay, but they all have it. They're all expressing. Yeah? Okay. Okay. So let's talk about potency. When I hear potency, I'm thinking potential. Okay. So the more potent something is, the more potential it has to become anything. Okay. Little way to remember, it goes multipotent, pluripotent, totipotent from least to most potent. And that's in alphabetical order. How great is that? So why do we need stem cells? Why are stem cells so cool? Why do we need different levels of potency? You're a little future doctor here. You want to explain? You want to explain?
So stem cells are basically cells that can become anything depending on their potency, right? So in medicine, say they're missing disc in between their spine and their vertebrae, right? A common thing is doctors will inject stem cells into it, and the stem cells will turn into that disc that they're missing, so it like regenerates itself. There's also stuff with artificially made organs. If someone needs a kidney, you can get stem cells and have those stem cells get together and make a kidney by turning into kidney cells, right?
Yeah, super important. We need them. Okay, let's talk about the different levels of potency. Totipotent, I think like totally, right? So totipotent has the potential to differentiate into any cell required for fetal development. Okay, and they come from the zygote. That's in capital letters there. That's where they come from. Pluripotent, it's the same thing, but you have to remember this asterisk. You have to remember this asterisk, okay? The difference is it can become anything except extra embryonic tissue. Let's all say that phrase together. Ready? Extra embryonic tissue. You're going to see this and you're going to have to write it, okay? Because I'm sure she'll ask you a question about what is the difference between this one and this one. There was actually one in your mini midterm. We're going to go over in a second. But basically, these are the same, but this asterisk is so important. Totipotent, totally, can become anything. Pluripotent, a little bit less, can become everything but. And remember that alphabetical order. So if we're going from least potent to most potent, starting with multipotent, is multipotent, knowing that it's the least potent one, is it the most specialized or is it the least specialized? It's actually the most specialized, right? Because if we're talking about potency, we're talking about the potential to become anything. We're talking about multipotent. It's actually the most specialized because it has the least potency, has the least potential to become anything. So that's why it's the most specialized. Does that make sense? Just think potential. That's what I like to think. Like the more potency something has, the more potential it has to become anything. Okay, so that's that multipotent one. So that's the only type of stem cells that adults can have. So that's more limited as into what they can differentiate into because they have more specific jobs to do. Does that make sense? Okay, so which one is the least potent? Multipotent, and which one is the most? And what can't pluripotent cells become? What can they turn into? Extra embryonic tissue. Great. Oh, you guys do so well. Okay. Part two. This is the greatest photo you're ever going to be given ever. Okay. This has it all. It has it all. When you see the phrase blastocyst, okay, all I want you to think when you see the word blastocyst is pluripotent. Okay. That's what I want you to think. Just think pluripotent. So the blastula here, there's a little difference that we're not going to get into, but when somebody says blastocyst on this exam, you're going to think pluripotent. They're pluripotent. Okay. So looking at the zygote, what level of potency do guys, the stem cells from there come from? Totipotent. Okay, that's the most. We can become anything. And we get a little bit less pluripotent, which means we can become anything but extra embryonic cells. And then we get more specialized. This is an adult. This is you. This is literally you right now. Okay, so getting a little bit more specialized. And then we have like specific types. Okay, and just a couple definitions: embryonic, fetal, baby, human tissue, extra embryonic, fetal support tissue, like the placenta. Just know that phrase. You don't have to get into the weeds of it. Okay, and finally, she didn't ask this on your mini midterm, but I am suspecting she will do this to you on the exam because she didn't talk about it. Okay, embryonic stem cells, ESCs. They are from the inner cell mass of the blastula, blastocyst, and they are pluripotent stem cells. Does anyone know, and if you're here on the first one, you'll remember this, why are they controversial? Why are they controversial? Yeah.
Because they use the actual, so they made embryos and then would throw them away after taking the stem.
Yes. So in order to extract these ESCs, which they're super helpful because they can help regenerate tissue, right? To get these guys, we have to like damage the embryo. That's why they're controversial, okay? So then instead, we have iPS cells. So we make these types of cells. We don't extract them right there, we're just making them. So we're just trying to get to as close as the ESCs as we possibly can without hurting the embryo, okay? That's the difference between those. We're making these ones to try to be like this without hurting an embryo, okay? They are less controversial than the ESC, less likely for the body to reject them if they're used. But here's the thing, is like we can never truly recreate the history of a living cell. We can't do that, okay? We can get as close as we can possibly do, but we can never recreate it exactly. Does that make sense? Okay, so if you talk about controversy or like what's the pros and cons of using each, I would center my answer around that thing. Yeah.
So for the controversial aspect, is it because we're hurting the ICM or are we like hurting the trophoblast?
It makes like the embryo non-viable. So you
Embryo is in what part?
like the fertilized egg. Like so you have
That's the zygote.
it'd be this. Or either of these, like at that stage. Like when the sperm fertilizes the egg and it has all the totipotent cells.
Okay, so from the blastocyst, we're taking out the ICM for the ESCs or?
Not at this point yet, because these cells right here are pluripotent, not totipotent.
Okay.
We want to have cells that can turn into anything. So just think about that it's controversial because you're taking the cells from an embryo and then the embryo is like non-viable. So it's like ending a pregnancy kind of. Makes sense?
And then, if we have time, can you go over what you would use, for specific examples? So I think in the problems that we talked about, for like organ transplant or something else, and which, kind of stem cells you use for them.
Which one are you talking about on the problem set?
Couldn't tell you, but we had questions about how like if it's a organ transplant, you're gonna use, unipotent, whereas if it's some other instance, you're gonna use pluripotent.
We can go over problem set stuff at the end, but we're just going to try to get through this for now because we got a lot of 2 and 3. But at the end, we're going to stay behind. You can like come up and we can go over it. Okay. Okay. Another mini midterm question. You can find totipotent stem cells in a blastocyst. Who knows the answer to this one? Any guesses? Any guesses? Thumbs up, thumbs down. Yes for yes you can. Thumbs down for no you can't. Okay, well let's go over this. So no. Okay, look at this photo once again. Blastocyst over here. Okay. Once we get to that stage, we have become pluripotent. Okay, yeah.
The inner cell mass cells pluripotent are the outer cell mass cells
The outer cell's mass would be totipotent.
Isn't that still considered part of the process? Like, there's like, is it two divisions of processes? There's the inner cell mass and the outer cell mass?
This is talking about just the blastocyst would be kind of just the inside,
That's what she's talking about.
Just break the inner cell mass up.
basically. Yeah, think about it like that. She wants you guys to think of it as separate,
Yeah, just
because
focus on the inner.
that outer part is the extra embryonic tissue.
Like, the whole, like, totality of it, like, because of the different divisions of the process.
You're like confusing blastula and blastocyst, but you guys don't need to know. Kind of, yeah.
But you don't need to know that. Just think, when you think blastocyst, just think pluripotent. Okay? Okay, let's do the next one. I can't even say this word. Something stem cells are an example of multipotent stem cells found in the adult body. Does anyone know what that term means? Anyone know what that means? Yeah, so it basically means relating to blood cells. Okay. When I see kind of like a specialized term like that, especially on this exam, it's like really specific, right? We talked about it briefly, but like I wouldn't expect you to like know that right away. But when I see a specific word like that and you're like, it's not the ESC, it's not the IPS, it doesn't say anything about potency, I don't really recognize it until it says multipotent, then I would kind of think it's a little bit more specialized. And more specialized means what type of potency? Multi. Okay, so that was the one I got asked about a lot in the exam because people were like, did we learn this one? Very, very briefly, she might have mentioned it, but when I see a crazy term like that we have not gone over, not the ESC, not the IPS, and it's telling me multipotent, I'm thinking that it's a specific thing. It's doing a specific thing. Okay, so that's how I would tackle that question. Okay, so it is true. So multipotent equals least potent. So it has the least potential to become anything it wants. So it's the most specialized, relating to production of blood cells. Here's the definition, okay? They are bone marrow stem cells that can become a more limited number of types of cells, like red blood cells and different types of white blood cells. That's just basically saying it's multipotent, which means it can become a limited number of things. Yeah. If there's something that you don't know yet, is it's multipotent? So I don't think she's going to give you another question exactly like this on the exam, but if I were approaching this question and I don't recognize it based off of the types that the specific ones that we've talked about, and she's throwing in the word multipotent in there, she's making it specific, and you're thinking about this one specifically we learned, like the ESCs and the IPS, and I don't recognize it, I'm going to assume that that's a more multipotent thing that's most specialized. Yeah.
Oh, I can do this and then you tell me.
Yeah.
All right. Is everyone all set with topic 1? If anyone has super specific questions, we'll stay after and we can answer those after. But we're all set. All right. All right. So basic amino acid structure, like we talked about before, they all have the backbone, the same backbone, right? That'll be the carboxyl group, the central carbon, and then the amino group. And the difference between all of them will be the R group or side chain. Okay, so this R group and side chain is what gives it all of its characteristics. Everything else is the same. Primary structure. So primary structure is the most basic. It's just a chain of amino acids together. So you literally just have amino acid, amino acid, amino acid. Do we know how they're connected? Go ahead. Covalent bonds called peptide bonds. So do we think that'll be between R groups? Do we think that'll be between backbones?
Any guesses? Anything? Yeah.
Backbone, exactly, right? So here you see 2 amino acids, right? This is the backbone, this is the backbone, and they connect right here. And you see water's being released in that process. And then eventually you'll get a chain like you see here, right? It could be any order. Order doesn't matter.
And what type of model do we show this with? So does he ask you like, how do I represent primary structure? What are we doing? Backbone model. You see, I was just like, literally like the least detailed thing you've ever seen. It's just like a bunch of zigzags. Do I see R groups? No, I'm just seeing literally like the skeleton, right? So that's why it's good for primary structure, because primary structure just means it's literally just the sequence of amino acids. Okay, so when you think of each structure, I want you to think of the type. I think she asked on the mini midterm as well, but I want you to think of the type of model that you're going to associate it with. And for some of these, there's going to be 2 of them. But for this one, it's just the 1, right? Because we have very limited information to put in this model.
What's a way we can change the primary structure? Say it out loud. Yeah, exactly. So if you change the amino acid sequence, say I take out this glycine right here and replace it with something else, the code is a whole different code. It's got a whole different amino acid structure. Even just if it differs at 1, that's a whole different primary structure. Does that make sense with everyone?
Because imagine you're changing the amino acids, you're changing the chemistry, which means you're changing the what? The structure, which affects the what? The function. Okay.
Is there a difference between the backbone model and the
Yes, we're going to talk about that.
Okay, secondary structure. So secondary structure is made up of 2 main structures. There's the alpha helices, which is like, think DNA. And then you have beta sheets, which are almost just like flat sheets that stack on top of each other. The difference between this and primary structure is that this is formed with hydrogen bonds between backbones of different chains. Here you can see this would be an example of the beta sheets. These are hydrogen bonds between the backbones, putting 2 sheets together. Here in the alpha helices, you see the hydrogen bonds kind of going up and down on the helix. What is a way, how could you change secondary structure? Think about, would changing an amino acid in it change the secondary structure? Where are the interactions happening? Backbone or R group? The backbone. Remember, these are backbone interactions. So if you change the amino acid, they all have the same backbone structure. So it wouldn't necessarily change secondary structure. What could we do to disrupt those bonds to change that structure?
I just have a question. What part is the backbone? Does it just the molecules at the top or
Backbone is everything except the R group. Every part of the amino acid except. So if you ignored this R group, all this is the backbone. So basically, to disrupt this, you need to add things that would disrupt those hydrogen bonds. So you could add heat, acid, like acidic stuff that would change the pH, anything like that.
And okay, so for ribbon model too. So when you see secondary structure, when she asks you types of structure that's represented in secondary structure, all I want you to think is alpha helices and beta sheets. That's all I want you to associate with secondary structure, okay, other than those bonds. Ribbon model, I feel like it makes sense. What am I seeing here? The what? Alpha helices. Then I see like the little, like this. Those are the what? Beta sheets. Who knows? Okay, so like let's take an alpha helix, okay? Where are those R groups going to face? How do those R groups face? Comparing everything else. They're facing outward. Okay. So in an alpha helix, alpha helices, they're facing out. Okay. The R groups are facing out. Do we know where the R groups look like on a beta sheet? Up and down. Okay. You see that shape, right? So R groups facing up here, the bottom, R groups facing down. Okay, you're seeing the R there, right? Up, down, up, down. Does that make sense?
To remember it, think in the helix, there's no space inside. It's a tight structure, so there's no space for all those big R groups to point inside. So that's why they all have to point outside. And then same idea in a beta sheet. If you see the first R group right here is pointing this way, that means the next one will be pointing the opposite way and it's alternating. So left, right, left, right. That's because if two consecutive ones are on the same side, they're too close to each other. They don't want to be close to each other. So that's why they alternate to have the most separation between each other. They're between two separate ones. Enter. Wait. Real quick, we'll go over it later, but does everyone get what a hydrogen bond is? Okay. So true or false, secondary structure is caused by hydrogen bonding between R groups. False, why? Exactly. It's caused by hydrogen bonding between backbones, not R groups. And then true or false, a protein with one polypeptide can contain multiple domains.
Any guesses? Yeah. Yeah, okay. You know what? Let's just go over that real quick. Okay, so do we know what a polypeptide is? Okay. Poly means what? A lot of things, right? So polymers, we're putting together things, okay? Polypeptide, peptide bonds. So what must that mean using those clues? What is it many of? Amino acids. Peptide bonds, link together amino acids, polypeptide, a lot of peptide, okay? Polypeptide. So bunch of amino acids, okay? And remember, they all fold a different way depending on the chemistry. So we take one string with a bunch of different amino acids. It's going to fold differently than if I take a completely different set of amino acids and string them together, right? Because they're chemically different. So a domain is an independently folded part of a single polypeptide chain. A subunit can contain multiple polypeptides, okay? Single polypeptide in a protein with quaternary structure, yeah, made up of multiple polypeptide chains. So I take one polypeptide chain and it has different folded parts, okay? Because why? Why does it have different folded parts? Because of that specific amino acid sequence. They're all going to fold different ways within this long chain. I put a bunch of those chains together. I can call it a subunit. And within that large subunit, there's those different polypeptide chains that are all together. And within those polypeptide chains are the what? Domains. Okay, so a lot of domains together. Domain just means like an independently folded part because a big string has different amino acids. There's different chemicals, so they're folding different ways. Put a bunch of those together. Polypeptide, just one. Bunch of polypeptides. We have a what? Subunit. Just think subunit is like just multiple of those polypeptides. Okay.
You talk about domains that you're talking about.
No. Domain is just a clump of the same protein, of the same chain. It secondary structure has nothing to do with it.
So you'll have like just think about like the sequence of amino acids, right? We have one polypeptide. We have one string of amino acids put together. Within there, I have like MET, MET amino acid. I have different types of amino acids in there. And like 4 of those together are going to fold in a certain way. And then I go to the next 4, and those are going to interact together and fold in a certain way. And I go to the next part, all in the same chain. So those are the different domains within the one polypeptide. If I get multiple polypeptides and I put them together, that's a subunit. Okay, so think subunit is the big thing. We zoom in on one particular chain in that giant subunit. They're going to be made up of domains. Okay, so just think like zooming in. Start out with subunit. Let's go to one chain. Within that one chain, there's going to be domains. Okay.
You want me to draw it out for them?
Here, I'll
So
take it for you.
how do you take that pen out? Shit's still.
We're gonna draw it out for you guys real quick.
All right, so think subunit. You have one chain. You could have it like this, and then there's a clump in here, and then it goes like that, clump here. You saw that was all one line, right? I didn't lift up the pen at all. That's one chain. There's one clump here. That's one subunit. One clump here. That's or domain, sorry. one clump there's a domain, and one clump here is a domain. Two domains, right? And now subunit would be if I added another one, another chain. This whole thing are two chains together. That's a subunit. Does that kind of make sense? It's hard to tell. There's not going to be a specific start and end. You most of the time you'll just notice different clumps. Sometimes, yeah, sometimes images will have different colors and they'll kind of color code it so it's easier to see. If it's different subunits, different chains will usually be different colors. So that's how you'll be able to tell it's different subunits. With domains, they tend to be the same color, but you'll distinctively see different clumps.
Yeah.
All right. Like a common one will be hemoglobin, right? It'll have like a chunk right here, a chunk right here, chunk right there, chunk right there. Right? So then you have the 4 subunits, 4 different chains together.
Yeah, so just think, okay, subunit, giant, huge thing, made up of different polypeptides. Bunch of polypeptides make up a subunit. Within the singular polypeptide, we have the domains. Within the singular polypeptide, we have those chunks that he just drew out.
I'll talk more about it here in this slide when I go over this.
I want to go back.
Yeah.
Okay.
All right. So tertiary and quaternary. So first tertiary. Both of these are the overall fold of a protein. Tertiary, the difference between quaternary is that tertiary is just one chain, right? So this can't have subunits because subunits is multiple chains, but it can have domains, right? So ignore this image for now because that's quaternary. But you'll have, like I talked about, you'll have one chain and it can be folded infinite ways. If that chain has different chunks within it, folded like in a distinct structure from each other, that'll be a domain, different domains. All right, but think tertiary, just overall fold of one chain.
And the fold depends on what?
What part of the amino acids? R groups, right? The R groups are doing interactions with each other. Do we know what types of interactions? Next slide. All right.
Okay.
wait.
Oh,
There's no slide on quaternary. So quaternary, the difference in quaternary is that it'll be multiple chains. All right. So same thing. R groups are interacting with each other. Same types of interactions that we'll talk about for tertiary. The only difference is that it's multiple chains. Okay. So not every protein has to have quaternary structure because some proteins will just be one chain. And then there'll be more complex proteins that are multiple chains and they'll have quaternary. But everything has to have tertiary because every protein is at least one chain that folds upon itself.
yeah. Every protein has an overall fold. Actually, stay on here for a second too. Okay, so let's talk about like the models real quick. So this, you can see it's color coded, right? So basically that would be a good representation for quaternary structure because it's showing us the different change and how they all fit together. If I took out, like if I just took the gray from here and I made that into like a space-filled model, would that still work? It would, because it's showing me the overall fold of the protein. So you could use that to represent tertiary structure. This over here, she kind of groups them together. Wireframe / ball and stick, those are two different models, but she's going to refer to them as the same thing. It's this guy. What information are we getting here with this model? I see balls and I see sticks, right? So what do the balls represent? What specifically? What is that key thing that are interacting? R groups. And what do the sticks represent? The bonds, right? So I'm getting a lot of information compared to that squiggly line from the primary structure, right? So let's go through them. Primary structure, what type of model? Backbone model, skeleton model, backbone model. Secondary structure, which model am I going to use? Ribbon, right? Because alpha helices, beta sheets. Tertiary, what am I going to use? And either of these work. So you could say, this might be a better way, sorry, the spatial might be a better way because I can see how much like space it's filling up. Like I can see the overall size of this thing. But you could also say, hey, I'm looking at the ball and stick and I'm seeing how the R groups are interacting and I'm seeing those bonds. So you see how there's pros and cons in each of these models, right? So we're starting with the least amount of information, which is primary structure, which is just the sequence of what? Amino acids. What are the two terms you're going to think of when you look at secondary structure? Beta sheets and what? Alpha helices. Tertiary, just overall fold. General, overall fold. Okay, and then quaternary, the bunch of those chains together. So it's just like you're taking this and you're like copy pasted a bunch of different sections. Any questions on that?
Let's say we take out just the gray portion. That's a subunit, and that would be tertiary structure, correct?
Yeah, exactly. So this whole thing is quaternary because it has multiple, each of these colors is a different protein. Well, a different part of the protein that comes together, a different chain that comes to make the whole protein. And each of those has its own tertiary structure because it's folded in itself. Everyone got that?
Secondary structure.
Yep.
The beta barrels included in that.
Beta barrels.
Beta barrels are a structure that are made by beta sheets. So it's when you have a bunch of sheets like together like that to make a barrel.
Okay, let's get into the interactions and the bonds. My advice here, don't try to memorize everything separately. I want you to think of it more like logically. So here's a chem breakdown. I didn't take orgo when I took this class. Everybody around me did, and I felt really stupid, but you don't need to know it. I'm gonna tell you everything you need to know for chemistry. Okay, don't freak out about the dipole stuff. We're gonna figure it out. Okay, first thing you need to know. Oxygen and nitrogen are more electronegative than carbon and hydrogen. Fundamental rule. Okay, I want us to say it. Which two are more electronegative than carbon and hydrogen? Oxygen and nitrogen. Remember that, okay? Electronegativity is just a fancy way of saying the more electronegative something is, like oxygen, the more that electrons are going to want to hang out there. So if I have an OH bond, where are the electrons more likely to hang out? At the oxygen, because it's more electronegative. Okay, remember, electrons are what type of charge? What do they have? Negative or positive? They have a negative charge. So if we have the OH bond and all the electrons are going over to the oxygen, what dipole is that oxygen gonna have? Negative dipole. Boom, you just learned chemistry, okay? And what dipole is the hydrogen gonna have? Positive, right? Because they're all leaving, they're all going to the oxygen. Does that make sense? Okay, so we know electronegativity, we know which ones are more electronegative than another. Another thing, polar. What? Water's polar. Hydrophilic. Hydrophilic means what? Water loving. Hydrophobic means what? Scared of water, right? Boom, easy to remember. Polar molecules, hydrophilic, I just think of water, I feel like that's not too hard to remember. Non-polar means hydrophobic. So we have the base rules, and let's get into the actual interactions, okay? So instead of trying to just like drive yourself crazy with memorizing every single one of these, I just want you to think like if I take one amino acid from one group and I compare it with another group, what's going to happen? That's it. That's this whole thing. And it all leads in with each other. So let's go through this together, okay? And we're going to go back to the chemistry explanation as well, so we can get both. When you're looking at polar here, don't just think polar. I want you to think polar uncharged. Why? Because we have polar charged over here. So these three are all polar. This one is polar uncharged. These two are polar charged. Okay? And that's all nonpolar. So let's take one random R group from here and nonpolar, and let's have it interact with another R nonpolar group. What is going to happen? What is that interaction called? Anyone know? We got this. We got this. It's like it's like a name. Hydrophobic effect, that happens. What's like that slash there? What is it? It's like that weird, it's van der Waals. Okay, so we know that oxygen and nitrogen are more electronegative than carbon and hydrogen, right? So if we have an OH bond, I know exactly where those electrons are gonna go. Oh, it's the oxygen. It's more electronegative. Let's take a CC bond. Is there much of a difference in electronegativity there? No, right? I can't say with confidence where the electrons are gonna wanna go. Okay, there's no dipoles really happening. So what happens when you interact two of those together, it's like the electrons don't really know where to go. Is that gonna be like a great bond if the electrons are just coming back and forth? Is that gonna be strong? No, because it can't make up its mind. So you have like a CH bond, you have a CC bond. That's why they're not as strong. Okay, that's why they're the weakest interaction, because there's not a difference in electronegativity as much there is if we compared an oxygen and a hydrogen. Does that make sense? Anyone have any questions on that? So when we're talking about like the strength of each one of these guys, I want you to keep that in mind. Okay, is there a difference in the electronegativity of the oxygen and the hydrogen? Yes, they're going to go to the oxygen. That means that oxygen is going to have a negative dipole because they're all going that way. Okay, so here. So we have that first one, right? Let's go through the second one. Polar charge, let's say it's a polar plus and a polar minus. What is that going to make? Word bank. Say it out loud. Ionic. Okay. To me, this one is the easiest to remember. It's like plus and minus, like a magnet, right? They're attracted to each other. Ionic bonds, are they going to be stronger or weaker than that van der Waal? Stronger. Because I know where those electrons are going to go, right? It's two opposites. That's pretty strong. It's like a magnet, right? So that's going to be stronger. Yeah.
How do we get a positive charge if they're both uncharged?
For the first one?
Yes.
They would. There would be no dipole there.
Uh,
Because there's not difference in electronegativity. We'll talk about dipole ion stuff in a second. All right, let's go on to the third one, polar uncharged and polar charged. So think either, for the polar charge, think either the plus or minus. I don't really care. What's going to happen then? Ion dipole. Why? Why? There's a big difference, yeah. The difference in electronegativity. So you're taking one totally boring polar uncharged group, boring, and then we have a stronger polar charged group. I know where that dipole is going to go. It's going to go to the charged one, right? But it has that much charge that it's like in a different group. Okay. All right, and nonpolar and nonpolar. We already did that. Okay, let's do polar uncharged plus polar uncharged. Did we already do that one?
No,
Okay.
this first one.
Yeah, the first one. From that word bank, what's that going to make? Polar uncharged plus polar uncharged. What's going to happen? What's going to be made? Word bank. Hydrogen bonds. Take one from the, go back to the chart. Take one from the polar uncharged. Remember, this is the polar uncharged. Take another one. They're going to make hydrogen bonds. Okay, so let's go through this real quick. What happens? What's the interaction called between the nonpolar and the nonpolar? What's that called? What's that weird name? Van der Waals / hydrophobic effect. We'll get into the hydrophobic effect in a bit. Let's go polar uncharged plus polar uncharged. What did I just say? What's that going to do? Hydrogen bonds. Plus and minus. Put those together. What's that going to make? Ionic. Okay, and then we have polar uncharged with either one of these. What's that going to make? Ion dipole. Do we understand the chemistry part a little bit better now? Does that make more sense? Okay, so don't try to memorize everything at once, okay? Everything leads into each other. Everything makes sense, and you don't need to have taken ordo to understand it, okay?
Did she tell you guys for the exam she was going to give you this, but without like non-polar, positive charge polar? What she said she was going to take away? Did she say anything to you guys
Well, they had it on the mini midterm. They had like the full chart.
with everything?
You guys had the full chart on the mini midterm, though, right? So yeah, so I feel
All
like
right, never mind that.
Don't-
Never mind. But here's a quick trick, so you don't have to keep looking back at the chart. If it has a lot of CHs or ends with a CH, that's non-polar, right? So look at all these. H, CH, CHs. This one has an SH, but it has a CH right there. All those are non-polar, right? Because CH bonds, non-polar. Only hydrophobic effects and van der Waals interactions. Positive charge, easy enough if it has a positive charge in the R group. Negative charge, easy enough if it has a negative charge in the R group. And that just leaves you with polar. Polar has to have an OH or an NH. All right.
Yeah, so think oxygen, nitrogen are polar. And then carbon and hydrogen are the non-polar guys. The more of each of those bonds between them, the more one way or another is gonna be. We're gonna go over one of the questions too from that mini midterm that talked about.
For the people planning on going to med school and taking the MCAT, you're gonna have to memorize all these, everything about them. 3-letter code, single letter code, structure, everything. So take advantage now and memorize them if you can.
Okay, but there's one that we haven't talked about. There's a special guy. I'm building a house, okay? I'm me, big back me. I'm building a house. And I want it to be super stable. I don't want it to fall down when there's a hurricane. I want something that's gonna give me super stability, okay? Thinking of in terms of that, disulfide bonds form when two cysteines are in close proximity. If you don't remember which one is cysteine, it's that special one in the nonpolar with the SH there. When I put two of those guys together in close proximity, it's going to give you a super stable bond. Okay, so she's going to give you a question that's like, all right, I have a sequence. I want to make it more stable. I want to increase the strength and like stability of this structure. You're going to say, I would recommend putting in some disulfide bonds. Why? Because they're super strong. So let's talk about the strength of each of these. Which one out of these is the weakest from the word bang? Weakest guy. Van der Waals. Why? Electronegativity, not much difference. Electrons don't know where to go. They're just floating back and forth. They don't really care. What would the next strongest one be?
It'd be hydrogen bonds, right? Because what type of bonds are hydrogen bonding? It's like two dipoles, right? You have partial charges caused by different electronegativities. It has to have a hydrogen, and then it could be anything more electronegative, a lot more electronegative than a hydrogen.
Like an OH.
Okay. Does that make sense?
So that's so number one, the least one, the worst one, Van der Waals / hybrid. We'll talk about the effect. Second one, return in terms of strength, getting a little bit stronger, hydrogen bonds. What's that next one going to be?
So if hydrogen's kind of like a dipole bond, dipole, what would be after dipole? No, so you guys are getting confused. Yeah, ionic would be after, but even before that, ion dipole, right? Because if we have dipole, and then ionic is stronger, in between would be when you have one ion, one side of it is an ion, one side's a dipole.
And ionic bonds, it makes sense that they're so strong because I think like a magnet, like plus and minus, like super strong. Okay, and then that last one is going to be what type of bond? What's the strongest guy? Disulfide. I'm building my house. I'm building my house. What? Question? Yeah, disulfide, super strong. And it's really special. Okay, it's so special. You know the what?
Cysteines.
Yeah, cysteine.
Cysteine.
So you take two of those guys, close proximity, boom. I got what's something in construction? I got walls. I got floors. Okay, so does that make a little more sense? Yeah. Yeah, that's on my next slide. Boom. Boom, I got it up here.
So polar uncharged is hydrogen because there's no charges, but polar molecules have dipoles. So, and we know that all the polar amino acids have hydrogens. So that's how it's hydrogen bonds. Polar charged and polar charged.
Plus minus.
Always, if you have two charges, ionic. And then if you have one charge, so you have one ion, and then one polar uncharged, a dipole, that's how you get ion dipole.
And where the electrons going to go in those two groups? Polar uncharged or the polar charged?
And
It
then,
makes sense, right?
and then if you have two nonpolar, that means they're not charged and they're not forming dipoles. That's when you get the hydrophobic effect or Van der Waals interactions. And then disulfide bond, easy enough. Disulfide, two sulfurs.
That's that S.
Yes, in this case, same thing.
Yeah, same thing. You could say that, but we'll know
There's
what you're talking
there's
about.
slight differences, but you guys don't need to know that for this class.
Okay, so quick recap. The more CC, more CH bonds, which means that it's gonna be more what? Hydrophobic or non-polar. The more OH, ON, CO bonds, the more blank it's gonna be. More hydrophilic, more polar it's gonna be, okay? Hydrophobic effect, let's talk about that, okay? Hydrophobic effect is when the proteins fold into the tertiary structure. What does tertiary structure mean? It's the overall what? Fold, overall fold. By concealing non-polar hydrophobic R groups while exposing the hydrophilic R group. So all of the stuff that hates water is scared and caving in, while all the stuff that likes water is interacting on the outside. Okay, that's all that means. Yeah, so hydrophobic core region contains non-polar side chains. Non-polar means non-polar, doesn't like water. On the outside, it's interacting. So we have this, and then it caves in because it's scared, and you have this resulting. Okay?
So this right here, this would be an example of primary structure, right? It's just the chain, and then it has its tertiary structure here, how it folds overall.
Okay, and that was just a quick summary. We're not going to go through that, but yes, we have the weakest to the strongest here. So we have van der Waals, dipole, because like they're both kind of going in different directions. You don't have to, I wouldn't say dipole. I don't think she's going to say that phrase specifically, but ion dipole, ionic bond, covalent. I would think of this more in terms of like the interactions, like what we just talked about, but this is like a summary. Any more questions?
This picture will be super helpful for the exam. So make sure you know everything on this slide.
Oh, yeah, and super quick, disulfide bonds are covalent. Covalent bonds are very strong bonds. Okay. Right. Yep. And then you can look at this whenever you want.
This slide's important too. Make sure you know this.
Oh, and who can remind me, what are peptide bonds? What do they bond? To connect amino acids. Amino acids and a bunch of amino acids make up a what? Polypeptide, a bunch of polypeptides. Big guy. Yeah, there you go. So,
Take the picture.
and again, we emailed these to you guys if you don't have the slides. All right, this question. Since I'm like the best person ever, I've already labeled all of these guys for you. We're gonna talk about it. We're gonna talk about all of it, okay? Because I know that this was freaking some people out. So the gray ones, hydrogens, red, oxygens, and then what are these guys? Like what are the dark ones? Carbons, right? A whole bunch of those. Let's get into that first part of it. So first, label one atom that has a positive dipole and one atom that has a negative dipole. Explain why you chose those atoms and why they have that dipole. Okay, real quick. What does it mean when I say something has a negative dipole? That means more of what's going to hang out there. More electrons are hanging out there, right? And remind me, which are the two elements that have more electronegativity? Oxygen and nitrogen, right? And then the positive is obviously the opposite because all the negative stuff is going somewhere else. You're left with a positive dipole. Who wants to try answering this question? Who wants to give me an atom that has a positive dipole? Yeah.
One of the hydrogens with the oxygen.
One of the hydrogens with the oxygen, right? So right here, that's an OH bond. Where are the electrons going to hang out? Oxygen or hydrogen? Oxygen, because it's more electronegative, which means more negative charge from the electrons going to the oxygen, which means the hydrogen has a what dipole? Positive, because all that negative charge left going to the oxygen. Yeah, great job. Okay, go on to the next one. Okay, do you think this molecule is soluble in an aqueous solution? This is the hardest thing that you're ever going to say, okay? Discuss where you think you find this molecule on a scale of very hydrophobic to very hydrophilic compared to other molecules and explain why. This tripped a lot of people up and I totally understand why. Anyone have a guess for where on the spectrum from like super hydrophobic to super hydrophilic would you find this? Yeah. Moderately hydrophobic. Okay, you're not wrong, but I'm looking for something else, right? So let's talk through it a little bit. I'm seeing a whole bunch of what kind of bonds? Yeah, like I'm seeing like some CH, right? CH, whatever. What are those types of bonds called where it's like CCCH? Non-polar bonds, right? The more non-polar something is, the more what it is. hydrophobic, right? I'm seeing a bunch of those guys, but they're not that interesting. I'm also seeing a lot of OH bonds. Yeah, there's not nearly as many as like the CCCH ones, but they're there. And those polar bonds are strong. They're important. There's not much interesting things going on with the CC and the CH because there's not much electronegativity difference, right? But you throw in a couple polar bonds, it's going to give the molecule polar characteristics. Okay, so you're right in saying that, yeah, it's not going to be fully soluble probably because you have so many of those non-polar bonds. It's going to make it pretty hydrophobic. But that also doesn't mean that you can just completely neglect the polar bonds that we see here. So the answer, yeah, everything's a trick question. The answer is it's mid to decently hydrophilic. But why is it not super hydrophilic? Because of what? It's all those hydrophobic bonds going on there. So yeah, it still has those polar characteristics. We're seeing the OH, those are really cool bonds. But it's not going to be fully soluble because we have so many of these CCCH going on. Yeah.
I wouldn't be like into or like into hydrophobic. Then has more
Polar parts outpower non-polar parts. So if it has like this ratio of polar to non-polar would be more on the polar side, just because the polar is a little bit stronger than the non-polar.
Yeah, polar bonds are just a little bit stronger than non-polar, even though there's like way more non-polar bonds here. Those polar bonds that are there still have a lot of strength, so it's going to give it polar characteristics. Okay,
And
it's a tough question.
And the fact that the polar bonds are kind of spread out. So you see they're at the end, they're at this end, and they're in the middle. That also helps. If they were all just on one end, then that would be more moderate, right? Like one of the phospholipids, how it has the head, the polar head, and then the non-polar chain. Yeah.
Okay, do we want to take a 5-minute break before topic 3, or we just want to jump into topic 3? 5-minute break. 5-minute break. Okay, 5 minutes. We can do 5 minutes. We're doing exam 2.
Hold on, it's gonna be up there. Careful.
Hey, where's the friend?
he decided that he wanted to go like become the minor consent. Okay, so actually he's waiting in the parking lot back there. I feel like I'm in the building. So I have like to get mine.
What discussion?
Yeah, I mean your discussion.
Oh, actually,
The best discussion. I think the best discussion. What,
But
we
see
have,
that- but see
we
that
have
doesn't-
Hannah.
But see, that doesn't make sense.
We've got Hannah, we've got
Because I'm
Yanko, and we've got me.
What's your
We don't
spire-
need the discussion?
What's your spy rating?
Wait, no, once you know my spire idea, then you're gonna know which, like, how to think.
just it'll be in order of Spire IDs.
Oh, I'm
You hitting it? What group are you? Spy rating.
Wow. Okay. Wow.
It's in order specifically.
I like how this is like the worst.
We getting our midterms back?
At the end, some people are like heading out, so they want to get those before they head out.
Yeah, not right now though. If you're staying till the end, at the end.
I'm heading out.
Yeah.
What group are you in?
O2 AJ.
AJ.
Goes up to J.
Yeah, that one. I think it's just the whole rest of it. And you know it's by already?
Yeah.
Okay, yeah. So you can just look through those that are by ID.
You want one of us to start talking about it?
Are you guys heading out right now?
Yeah.
Okay.
I'm not heading out, I just want to.
Okay, if you're gonna stay here, would you mind waiting until the end, just because we're about to end the break?
Thanks, guys.
What discussions are you? Or do you?
I actually took two.
Oh, yeah,
AB?
yeah, thanks.
Nah, it'll be in order of Spire ID.
AJ.
AJ. AJ.
Yeah.
So if polar bonds are stronger, then why are they dissolved in water?
Because water is also polar
Oh,
and
okay.
light dissolves light.
Okay, got it.
What discussions are you guys in?
Okay, we're gonna start in a couple minutes. So if you're staying for a little bit, we're gonna have these at the end. These are just for people who are like heading out. I had no idea that you were.
I didn't realize you were one of the TAs until I handed you my quiz the other day.
Yeah. Which one?
O2AA.
AA. I'm gonna look through those. Spice right here. Which one are you guys in? AA.
Wait, I have color.
C. That's mine. Okay, AE. And then I need AC. Okay.
This is O2A. Thank you.
Okay. Want me to start?
Yeah.
Okay.
Let me put you.
Yeah, okay. Yeah, would you guys mind taking the piles just over there on that table?
Oh, yeah.
Okay, just in a matter of time. We're gonna put the exams over there. Yeah, take it over there. But we're gonna keep talking. So let's keep it down because we're recording this and it's gonna capture everything you're saying. Okay, so if you are coming up here, please try to go around so you don't walk in front, just so it's less distracting. Okay, DNA, RNA structure, transcription, RNA processing. Nucleotide structure, guys. I had to hit him with the teacher like. Okay, remember, DNA, deoxyribose, think delete oxygen. Okay, so if I'm looking at the structure of each of them, if I'm deleting oxygen for the DNA, that's what the sugar is going to look like. Guys, if you're collecting, just try to keep it down. Everybody just got theirs, please keep it down. Okay, so this is the difference between the two of them, right? Deoxy, delete oxygen on that second carbon. I have an H. RNA, just ribose, right? So it has that oxygen there. Again, we're kind of going to go through this part. Boston sugars in the phosphate groups. So this is the phosphate group, right? The yellow. That bond between them are called phosphodiester bonds. Everybody say that. Phosphodiester bonds. Big word. Okay? Bonds between nitrogenous bases to another base. So bonds between nucleotides are called what? Hydrogen bonds. And they are non-covalent. So super quick going through that. RNA, we already talked about this. RNA has the U, while DNA has the T. Okay? And if you remember from that problem set, that question, this is really important. The base pairing of G and C. If I take a G and I pair it with C, it's going to have 3 hydrogen bonds versus if I do an A and a T, it's going to have 2. So which one of those, if I compare a base pairing of G and C and A and T, which one of those are going to be harder to break? The G and C. So if I look at a whole string and I count way more GC pairs versus the AT pairs, it's going to be a pretty strong string, right? If I have like 6 GC pairs and I have 2, it's going to be pretty hard to break that strand apart because it's got so many hydrogen bonds. Okay, that was a problem on your question on your problem set, but just make sure that you know that GC is how many bonds? 3, and then AT is what? 2. Okay, and let's say I have more AT bonds between 2 strands versus the GC. Is that going to be easier or harder to break? Easier, right? Because it has less of those hydrogen bonds. No. Okay, let's talk about directionality. 5N and 3'N. This is confusing everyone. I promise it's not as hard as you think. Just remember that DNA prefers a double strand. RNA prefers a single strand. Remember that mRNA that's created from that one template strand, which we'll talk about. RNA can base pair with RNA DNA, but it's not as stable. It will never be as stable as the DNA in DNA. Okay, just remember that. But we're going to talk about directionality.
RNA cleavage from the 2' hydroxyl.
Exactly, right? Okay, so real quick. When you're approaching these questions, there's gonna be a lot of bases, and you're gonna be really scared, and you're gonna like panic. No need to panic. Remember this fundamental rule, and nobody will be able to shake you or trick you, okay? RNA polymerase is read from the 3' to 5' direction. If I'm RNA polymerase, I don't care if I write it, right? I'm gonna draw it a little bit. Can you see that? Okay. So let's say I have a strand that looks like this. So that's 3' A, T, G, C, A, whatever. First off, what type of, is this DNA or RNA? DNA, why? Because I see the T, right? I'm RNA polymerase. I know that RNA polymerase reads from the 3 to 5 direction. So I'm RNA polymerase. I'm gonna latch onto here, right? Because I'm going from 3 to 5. Now, how do I make that mRNA strand, right? And we'll talk about template strand, non-template strand. Don't think about that right now. Let's just assume that this is the template strand, okay? If I'm going through this and I'm making the new mRNA strand, because remember, that's what RNA polymerase does, right? It changes stuff from DNA to mRNA. I'm going to start here at 3. What in mRNA matches with A? U, right? Not T, because we're talking about RNA now. This is going to be the mRNA strand. Give me U. Now, T, shout it out. A, G, what? Which one? And, all right. And here's how you're going to label the ends. That's it. That's it. So I don't care if I see a strand that's labeled like this versus if I see a strand of DNA that's labeled like this. If I am RNA polymerase, all I care about is going from 3 to 5. That's all I care about. So I could take this and somebody tell me, if I'm just focusing on this guy, am I going to go right to left or left to right? Right to left. Why? RNA polymerase goes from 3 to 5' always. I don't care how I turn it in space. I don't care if I trick you and I write it backwards like this. I don't care. All I care about is reading it 3 to 5. Okay, so don't let that trick you. We'll get into some questions, but that is such a simple rule that people forget about because she's going to write it different ways and it's going to trick you. Okay, so somebody asked this question. RNA polymerase reads the template strand in the blank direction, but polymerizes the new strand in the what direction? What's that first blank? 3 to 5 polymerizes it in what direction? And remember, it's not like this crazy process. It's just going along, making it, and then that new strand, because it's the opposite, is just labeled the opposite way. Understand? Okay, yep. 3 to 5, 5, 3. Okay, Chargaff's rule. This was that percentage thing. I don't know if we're gonna go over that, but it just says that if we're complementary base pairing between 2 DNA strands, for every A, there's gonna be a what? A T, and for every G, there's gonna be a what? Right? So if I'm looking at the compositions of those 2 strands, I'm gonna look for an equal amount of A and an equal amount of T, and then an equal amount of G with an equal amount of C. Okay? All right, let's go through the process. Polymerase, RNA polymerase is going to make the transcript of the gene that it wants. The introns have to be removed. And then that 5' cap and that 3' poly A tail must be added for stability and protection. We're going to talk about that. And the mature mRNA finds the ribosome. You want to talk about that?
So if you look, when the introns are removed before we have that final mRNA, right? So what step would that be in? Transcription, right? This is during transcription before we have the final mRNA, because We need that final mRNA to be ready, like 100% what we want it to be before it's translated. Makes sense? All right.
Okay.
So we have the template and the non-template strand. Think about template strand. Yeah.
This might sound like simultaneously, would you say it's during transcription, or is it kind of before, or is it kind of after?
So yeah. So what transcription is the process of DNA to the final mRNA. Okay. So in that transcription, we're going to have the DNA turned into a pre-mRNA transcript, but then that transcript is spliced into the final mRNA. So it's like right at the end of transcription. Okay, template strand and non-template strand. Template strand is what serves as the template. Okay, that's what is being read by the RNA polymerase. And it has it's the template. So it has the strand that's being read and translated. But since it's being read and translated, that means it's going to have the complementary bases. So it's not going to have the exact bases as the DNA or the mRNA, because it's like the opposite of each base. Does that make sense?
Here, I can type on the next slide. Here. Okay, so if I just give you two strands and I label both them and I don't tell you any other information, do I know what the template strand and non-template strand is? No. Okay, so don't confuse yourself by looking at the two strands and just thinking, oh, well, the 3' is on the left and the 5' is on the right, so that has to be the template strand. That's not how it works. To find what the template strand actually is, we need a little bit more information. We need information about either the direction that RNA polymerase is reading, like right to left or left to right. Remember how I just asked you that question? But we know that it reads 3' to 5', right? We know that. And we also might need to know where the promoter is. Where is the promoter? Where is the promoter? Upstream of what? The what prime? 3', right? Because RNA polymerase reads from what to what? It needs to know where to go, where to start. We know that direction. If we know where the promoter is sitting, we can guess what direction that RNA polymerase is going to go. Does that make sense? Okay. Or we need info about the mRNA resultant strand. So that one on your problem set, that was like, which one is the template strand? If the RNA polymerase reads it right to left or left to right. Now we can figure that out because we have that information about the mRNA strand. If we translate it, if we already have that, we can work backwards. We're going to go over a couple questions, but just remember, you can't just look at the two strands and automatically know which one is the template and which one is the non-template. That's not how it works. You need more information. Yeah.
Is the promoter for the template strand 3' to 5', or is it on the non-template strand?
It's on the template strand, right? Because where are we making the mRNA strand from? The template strand, right? So we have the template, the non-template. If I'm RNA polymerase, I don't really care about that non-template strand. I just care about that template because that's where I'm translating it into the, transcribing it from the template to the mRNA. Yeah. Is the promoter on the 5' only for the mRNA?
The promoter's on the DNA.
It's on, so listen. Promoter tells transcription factors to latch onto that. The transcription factors tell the RNA polymerase to start there. What direction does RNA polymerase read from? What end to what end? Right, so we're starting at the 3, we're ending at the 5. So if I'm a promoter, am I going to want to be closer to that 3 or the 5? 3, right? Because that's where we're reading it from. That's the direction. Starts with the promoter, transcription factor latches on, RNA polymerase latches on, then it starts doing its thing. And you know for a fact that it reads it from 3' to 5'. Yeah.
Sorry, just go back to the last slide.
Yeah.
For which strand isn't read for the DNA, it's the coding strand.
Yeah, so that's a non-template and coding strand. I'm going to tell you a little secret. Okay, there's two strands, right? There's template and there's non-template, and then you get the resultant mRNA strand from that template. If you want a shortcut, because you're like me and you're lazy, if you go to the non-template strand and you replace all the T's with U's, That's actually what the mRNA is. Shortcut, because I'm lazy, okay? So again, you're gonna write that new strand in the 5' to 3', right, still? But if you just change this, the T to a U, T to a U, because that's the difference between DNA and RNA, you got your mRNA strand. You don't even have to go through like the whole, I have to do that because I overthink and I'm like too afraid that I'm gonna mess up, so I go through the whole thing. But if you are confident in yourself, if you just change the T's to the U's, you have the mRNA strand. That's why it's called the coding strand. Because you think about it, you're like, wouldn't the template strand be called the coding strand because we're taking it there?
But we just don't, sorry, we just still switch to 3' and 5'.
Yes. So still remember, like, it's gonna be in the opposite direction. Any other questions on that? So remember, if you look at two strands, let me get 2 seconds. If you look at the two strands, you can't know what the template and non-template strand is right away, you need more information. But if you know fundamentally that RNA polymerase goes from 3 to 5', you're good. Yeah, what's the question?
Is the promoter not part of the double-stranded DNA?
So it's part of the double-strand DNA, but it's on one of those strands because I'm only using one strand to make the mRNA,
Okay,
right?
and then you talk about transcription factors in RNA polymerase having their own directionality. How is that possible?
Own directionality. They're all going, their promoter is letting the transcription factor latch onto the promoter itself. It's not going anywhere, just latching onto the promoter. That is what tells RNA polymerase to go 3 to 5. So they're not all going in separate directions. It's in one place. The promoter is latched onto one part of the gene. The transcription factor latches onto that one part of the promoter, which then tells RNA polymerase, hey, you're going to start here and you're going to go in the 5' direction.
Okay.
So promoter, transcription factor, they're not moving. They're just latching on to a part of the DNA. The RNA polymerase is the only thing that's actually moving.
No. So we talked about how the transcription factors and the RNA polymerase have directionality itself.
What do you mean by directionality with the transcription factor?
Really quote unquote from Laura, I'm not sure.
Quote unquote from Laura. Okay, well.
It just means they have different
They have
direction.
One direction.
Yeah.
They-
So we know that the RNA polymerase goes in the direction it goes.
That's
Is
it.
there like a structural component to that or not important?
Not important.
Just think promoter is stuck onto a part of the gene. That's how we know which part of the gene to transcribe. The transcription factor latches onto that promoter to tell RNA polymerase, hey, we're going to start here and we're going to go in the 5' direction.
And if we choose, if I am RNA polymerase and I choose the top one as the template, I'm gonna get a different strand than if I use the bottom.
Yeah, okay, so like that was an exact question from your problem set, right? Like if I just choose a different strand and I try doing the same thing and it's not the template strand, you're going to end up with 2 different strands, right? Because why? Because if I'm reading from 3' to 5' here, again, RNA polymerase always reads from 3' to 5', and I try doing the same thing, but in this direction, so it's a different strand, you're going to come out with 2 completely different things.
In cells, though, there's a way that the cell knows which one is the template and which is the non-template strand. Yeah. Uh-huh.
And that's part of the transcription factors too, right? So it senses that things need to be done, and that's what gets RNA polymerase over there. Make sense? Yeah. Is the promoter more towards 3'? Yeah. So again, we're always doing it 3' to 5'. If you remember that, you know where everything else is. Okay. Okay, let's do this real quick. You are studying a particular gene, blah, 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 and you see this. Which strand for the DNA is the template strand? And how do you know? Explain your reasoning. Just starting off. Does mRNA, is it made from 1 strand or 2 strands? 1. We only care about the template strand. That's all I care about. There's 2 different ways you can approach it. Who can tell me which strand they think is the template strand and why? Yeah.
I think it's DNA strand 1.
DNA strand 1. Okay, why?
I like that.
Right?
So,
What else? More evidence. Your Laura's exam. You got to spit out everything.
yeah, if it's identical, like strand 2, or pretty close to it.
Identical and like there's the same amount of A's in both of them.
Yeah.
Okay.
And so if they're like pretty similar, then it would be
Done.
non-template,
Non-template, which is the coding. Okay, right? That's one way to think about it. Another way to think about it is if I'm going from one strand and I'm making the mRNA strand, I'm going along looking at the A's. For every A in the mRNA strand, there has to be a what? A U. For every G in the DNA strand, in the mRNA strand, there must be a what? A C, right? So don't look comparing the number, like for me, it makes more sense to think about it like for every A, I know in the mRNA strand, there has to be that similar number of the one that base pairs with it. For example, A matches with U in mRNA. So I'm looking at A, just going DNA strand 1. A, I'm going over to U. Oh, that's a pretty similar number. And they base pair, interesting. Going to G, what pairs to G in mRNA? C, let's go over to C. Huh, pretty similar number. C pairs with what? G, right? Like you kind of get the point by now. But let's say I didn't know that and I'm starting off with DNA strand 2. I know for every A, there has to be a U in the mRNA strand. If it's the template strand, we don't know which one it is yet. For every A, there must be a U. 24, 19. Sus, I'm sus. G, sorry. Yeah, G, looking at C here on the mRNA. 26. I'm sus. Okay, does that make sense? So that's what you have to think about it. These are simple things. For every A, there has to be a U in the mRNA strand. So you start off with 1, the template strand, I'm RNA polymerase, I'm going from what end to what end? 3 to 5. I'm latching onto that 3. And so for every A that I see in that DNA strand, I'm going to spit out, if I'm RNA polymerase, I'm spitting out a what? A U. And for G, I'm spitting out a. Any questions on that? So somebody just confirm for me, which one is the template strand? 1, right? For every A, I see a U. For every G, I see a C. They're not going to be the exact numbers, but they're similar enough. Got it?
And if 1 is the template strand, what does that make 2? The coding or non-template strand. And remember what we talked about, the little cheat, the non-coding or the coding or non-template strand will always be the same as the mRNA. So you guys can see here, both have similar A's, both have similar G's, similar C, and
Just
then
replace the T with
T
U.
and U, similar. Okay,
Yep.
this is what we talked about.
Okay, real quick, just to confirm. So if your coding strand has a sequence 5 prime, blah, 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 to 3 prime, in what direction would the RNA polymerase be moving if we know that's going from 3 prime to 5 prime, always? From left to right or right to left? Right to left. Right to left. See how simple it actually is? Okay, what is the sequence of the template? Let's go through this here. I'll write it down for this one to work through it. Okay, so it's the strand. Can you guys see that? Can you go back? So it's 5 prime, A, G, T, A, C, G, T, A. And if I'm RNA polymerase, I'm starting at what end? Starting here. All right, I always label my ends first so I don't get confused. If I'm RNA polymerase, I'm going along. I'm spitting out a what? U, right, because it's mRNA. Spitting out this, A, blah, blah, blah. You guys get the point. Okay. Okay, let's take that same strand and let's make a complementary DNA strand. How am I going to label my ends? Opposite, so this is going to be a what? 3 prime. This is going to be a 5 prime because they're complementary to each other, double-stranded DNA. Once again, how is this going to look? What should I do? How should I do this? How should I work backwards? Okay, I'll run through it with you. Same thing, I'm just tricking you. You're just going along. It's just the opposite. It doesn't really matter what direction I go in because this is DNA and we just know that they're complementary to each other. Okay, if it's mRNA, then it matters if I'm reading it from the 3 to 5, right? If they're complementary DNA strands, I just know that they're the opposite of each other. Cool? Any questions? Directionality? Cool.
All right, promoter and terminator sequences. Like we talked about, the promoter is at the 3 prime end of the DNA. The function of that is basically where the transcription factors will bind, and they'll tell the polymerase, this is where you need to bind, this is where you need to start transcription. Everyone got that? All right, terminator sequence is similar, but that's where it tells it to stop. Okay?
Yeah, and you'll get more into that in like topic 4, but that's all you pretty much need to know.
Then we have RNA processing. So once we have this mRNA, it's in the nucleus. It needs to be brought out to the cytoplasm for translation. And before this is done, we need to add a 5-methyl cap, which is basically just a guanine nucleotide at the 5' end, right here. And that guanine has an extra methyl group than a regular guanine. The purpose of that is to protect the mRNA from degradation. So from getting chewed up and broken down by the nucleases that are floating around. RNA guanylyl transferase is what puts that guanine residue on, and it uses RNA triphosphatase and methyltransferase to put that on. Then at the opposite end, at the 3' end, we have the poly A tail. That is just a chain of adenine nucleotides at the 3' end. It has the same purpose. It protects from degradation, but this is also like the GPS that helps it get out of the nucleus. So without the poly A tail, the mRNA can't leave the nucleus to be translated into protein. Poly A polymerase is what puts the poly A tail on, but before that, CPSF is what cleaves the mRNA to add that tail. All right. So important things to remember: 5' methyl cap, 5' end, guanine with an extra methyl, protects against degradation. So protects the strand from breaking. 3' poly A tail, 3' poly, multiple A adenine. All right. Again, that also protects from degradation, so from getting chewed up, broken down. But that one is also the code to get out of the nucleus into the cytoplasm.
So they both protect from degradation, but one of them, the poly A tail, is important for even getting us out of the nucleus. Like think of the nucleases like little Pac-Mans. And if you like leave it open, if you don't have the 5' cap and if you don't have the tail, they're just going to chew away at that. And we don't want that.
Did you care about poly binding proteins?
She won't ask you about that.
And then that cleavage is cuts off the termination.
Yeah. And we'll talk about that last question on your quiz that has to do with this. Okay. Actually, should we just do that right now?
Yeah.
Okay. Let's just do that right now.
Yes or no?
For what?
You'll see. Okay. Oh, we love this question. We love this question. Okay. For any gene that contains introns and exons, if a 5' splice site, what does splice mean? What does splice mean? If a splice time for when the introns were mutated, would the gene still be, yes or no? Briefly explain. Think about what controls these processes. Make sure that information in your answer and whether a 5' splice site mutation would affect that process. So before I even think about introns, exons, whatever, transcription is just going from what to what? DNA to mRNA. Okay? To the final mRNA. Remember what Yago said. So there's still RNA processing going on in there to become the final mRNA. Does anyone have an answer for this? And why? Yeah.
There's like a mutation in the exon, it's still gonna get like cut out.
Okay, let's go over it. Yes, so the answer is yes. The gene would still be transcribed. It would still be transcribed. So that's saying that this mutation doesn't have an effect. It's saying that it would be transcribed. Mutation of the 5' splice site wouldn't prevent transcription from occurring because transcription is controlled by RNA polymerase, right? So we're talking about the splice site. That's way later. first we need the RNA polymerase to actually do the transcription. Okay, controlled by RNA binding to the promoter region of the gene, not by splicing. So by splicing, that's basically saying that it's happening after I've already made the mRNA, right? So let's go back to that photo. Oh, go back to that photo, right? Where is it? So transcription, before translation, it does involve the RNA processing, okay? But before the RNA processing, we still get a strand of that mRNA. So by having the splicing and stuff happen over here, that doesn't really affect up here where that RNA polymerase is actually doing the transcribing. It's going to affect the final RNA, right? The pre-mRNA. It's going to affect that final guy. But for the actual act of transcribing, that first step before we even do any of that, it's not affecting the RNA polymerase. Any questions on that? Okay. Let's go back. And now, what we just talked about, given a poly A tail. So we're saying that we're mutating this 5' splice site. And it says, would the gene still be given a poly A tail? Answers. There's a mutation. Something's wrong with that splice site. Who wants to give an answer real quick? Yes. Okay. Why? 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 Okay. It's late. Let's just go into it. So the gene would still be given a poly A tail because, okay, celebration. Okay, cool. The addition of the poly A tail occurs after transcription during the RNA process. So, quiet down, children, quiet down. So again, transcription happens first. So the RNA polymerase is going along transcribing. Okay, are we going to adjust the mRNA and all that stuff before we make the final mRNA? Yes. But transcription is just that first part, taking that template strand and making that like first initial mRNA. Okay, yeah.
If we go through the process, you would do transcription,
Yep.
5' cap, poly A, then intron splicing, because 5' cap is automatic as soon as it gets out of the RNA polymerase. And then we have, if the intron splicing didn't affect the poly A, then it would be polyadenylation, then splicing.
So again, so what if I said, instead of given a poly A tail, what if I said the 5G cap, given a 5G cap? Would it affect it? Yes. We're messing with that 5' splice site. Okay, so if I change this to the 5G cap, yeah, it's going to be affected because we're messing with that splice site. What does splice mean?
5' site of a intron, not the 5'.
It's A5 for one. I think that means though, like, because they're being spliced together, like it still needs that protection, right? If the answer was like, if it was a 5G cap, would it be effective?
Let's say it was in the middle of the coding sequence, so taking out the 5' life site of the intron, not the 5' ends, which is involved in M7G cap. Is that correct?
I think you're thinking too much into it. I don't think you need to know that much. Like same, even with the like specific order that you were talking about, you don't need to, like,
She's not gonna
don't
ask
have to
about the
worry
order.
about specific order. Like if you think about all those happening, similar timing, you'll be good.
That's exactly what he said.
Well, this one's not so much about the order. Like, did you write down the explanation I put? I sent you? Just follow this part. Can you see? This answer.
that question. Okay. Well, we're gonna go over the 5G cap a little bit more about that, like right now. Just to kind of clear things up.
Okay. If there's not a poly A tail, what do we think will happen? Think specifically, what's the purpose of the poly A tail?
Degradation and what else?
And localization to get out of the nucleus. So if we don't have that, it'll
No
be
GPS,
degraded and it won't be able to get out.
which means it can't do what? What's that process? Yeah, it can't be translated.
Oh,
That's the
yeah, hold on. And then do prokaryotes need a 5' methyl cap and poly A tail, and why or why not? Exactly, they don't need it because there's no nucleus, so it doesn't need to leave the nucleus, which would be part of the poly A tail's job. And then since both of these are happening at the same time, there's a shorter lifespan of the mRNA, so it doesn't need to be protected as much. Yeah.
Yeah, right? Because remember, prokaryotes, they do translation and transcription in the same place. Unlike eukaryotes need to be able to get out to the cytosol to do translation, right? Which is why we need the sequence other than just like protecting us from degradation.
They're just protected because of the simultaneous.
I don't think they have nucleases, but I don't think you need to know that.
Okay.
All right, splicing. So what you need to know about splicing is that introns are spliced, exons are kept. So think exons expressed. They're kept and they're expressed eventually. Introns, opposite of in, is out. They leave, they're out. What does that is a spliceosome. Easy enough to remember. Spliceosome is in charge of splicing, which is cutting. And then SNRNP is the protein that's going to base pair with the intron start sequence and cleave it there. Make sense, everyone? All right. You're looking at a bacteria and you want to see the effects of intron splicing, but you don't see anything. Why? Exactly. Splicing doesn't happen in prokaryotes. Bacteria is a prokaryote. It doesn't happen because prokaryotes don't have the machinery. So they don't have the spliceosomes and all that. All right, alternative splicing. So we know exons are kept. These are in red. Introns in green are cut out. Remember, exon expressed. Intron, opposite of in, out. So just because exons are kept doesn't mean they're always kept. Exons are what can be kept, but they're not always kept. And the reason for that is because we can get, if we have 4 of these exons, we can make different codes, which would code for different genes. So an example here, we could have 1, 2, 3 by cutting out 4. So that would make 1 protein. And then if we cut out 3, we could have 1, 2, 4. Those are different codes, make different proteins. I don't know.
There were different proteins. Yeah.
This is like one of the last questions for our problem set in 4.3. Does it always have to have the first?
I'll talk about that. We have a question on that. All right. So remember, purpose of alternative splicing, just to make different variations. Okay. Which of the following is removed during RNA splicing? Introns, remember, opposite of in, out. Exons expressed. Where does the RNA splicing occur? Nucleus. Because this happens before we have the final mRNA that is brought out. into the cytoplasm. Make sense? Those are the answers. What type of RNA undergoes splicing? mRNA. mRNA is the code RNA that starts in the nucleus and then gets brought out. tRNA is the RNA that works on the ribosomes that brings in the amino acids. rRNA is the RNA that makes the ribosomes. Okay? What does alternative splicing allow for? A, right? You have a single gene and you can have different variations of that gene to make different proteins. Okay. If you have an mRNA strand that has 4 exons, so exon 1, exon 2, exon 3, and exon 4, how many variants can you make? All right, ready? These are the possibilities. If you see all of them always have 1 and 4, right? Each of these has 1 and 4. 1 is always needed because that has the start sequence, right? You need somewhere to tell it to start. And then 4, since it's the last one, has the terminating or stop sequence. That's always needed also. All right. So what I did here is I wrote 1 through 4 for all of them. And then what can change is the 2 left, 2 and 3. So this one, there's no reason why it has to have either of those middle ones, as long as it has the start and stop. So this one has the start and stop with none of the middle, start and stop with one of them, start and stop with the other one, and then start and stop with both. Everyone got that? All right. And that's it.
We'll hang around like for a little bit. It's really late.
If you want to get your mini midterm, they're all up there organized by discussion section and Spire ID number.
Yes, 1 second.